Welcome. Welcome to the deep dive. Imagine for a moment a universe unfolding right beneath your nose, a vibrant, bustling metropolis of life, so tiny it's utterly invisible to our naked eyes.
It's everywhere, isn't it?
Exactly. Yet this hidden world profoundly impacts everything from how healthy we are, to the food on our plates, to even the very air we breathe. It kind of begs the question, how on earth do scientists even begin to study something they can't even see?
Yeah, it seems impossible at first glance.
Today, we're plunging into a deep dive on microbial cultivation, a truly foundational, often unsung process that allows us to not just observe, but to actually grow, study, and ultimately understand these microscopic inhabitants.
It's bedrock, really.
We've sifted through a stack of fascinating insights on the specialized media, the ingenious methods, and the clever isolation techniques that make this invisible exploration not just possible, but, well, deeply impactful.
Our mission today is to pull back the curtain on the intricate processes involved in growing microorganisms in a controlled laboratory setting, what we call in vitro, when they originally came from their natural environments. Or in vivo.
Right, taking them out of the wild, so to speak.
Exactly. This journey will reveal the meticulous dance required to separate and study pure cultures, providing you with a rapid shortcut to being truly well-informed about one of the most crucial aspects of modern microbiology.
Okay, so to get us started, let's maybe establish some common ground definitions. When we talk about introducing microbes into a carefully prepared nutrient material, we call that initial sample an inoculum.
Right, that's the starting point. Yeah, the little bit you add.
And the microscopic organisms that successfully take hold and multiply from that inoculum. That's what we refer to as a culture.
The resulting growth, precisely.
The entire intricate process of nurturing and growing them in a lab. That's cultivation. It sounds deceptively simple, but I guess it's profoundly powerful.
Oh, absolutely. There's a lot to it.
So why should you, our listener, care about how a tiny bacterium is grown on a dish in a laboratory? I mean, it sounds maybe a bit niche.
Well, you'd be surprised. Because this seemingly arcane foundational science underpins almost everything critical in modern biology and medicine. Seriously.
Like what, specifically?
We're talking about diagnosing infections accurately, developing life-saving new antibiotics, ensuring the food you eat every day is safe, and even advancing our fundamental understanding of life itself from its origins to its potential on other planets.
Wow, okay. So, big implications.
Huge. Get ready for some genuinely surprising facts and more than a few aha moments, I think.
All right, let's kick things off then with the absolute bedrock of microbial growth, the culture media itself. If you think of microbes as incredibly diverse, tiny organisms,
Which they absolutely are,
then you can imagine their dietary needs are just as varied. This media is essentially a carefully crafted menu or a specialized habitat, meticulously designed specifically for these microscopic guests.
You've got it. It's far more than just dirt and water. It's a precisely balanced nutritional buffet tailored to invite and encourage specific forms of life, sometimes very picky forms of life.
Picky eaters, even at the microbial level.
Oh, definitely. What's truly fascinating here is that bacterial cultivation is so much more than simply growing bugs. In a clinical laboratory and in research globally, it serves multiple, incredibly critical purposes, each building upon the last.
Okay, like what? What's the main goal?
Well, the absolute primary goal, the one that probably first comes to mind if you imagine someone getting sick, is isolation. We simply must grow and isolate every single bacteria present in an infection specimen.
Why every single one? Isn't there usually just one culprit?
Not usually, no. Clinical samples are almost never clean. They're a complex mixture of many different types of microbes. Your throat, your skin, a wound. They're ecosystems. To definitively figure out what's actually causing the problem, you need to unscramble that mixture and separate out the individual species into pure cultures.
Okay, that makes sense. So if you've got, say, a dozen different types of bacteria in a patient's sample, you can't just guess which one is the bad actor. You need to pull them apart, one by one.
Exactly. You have to isolate them. And that leads directly to the second crucial purpose, differentiation. Once you've got them separated on a plate, cultivation allows us to distinguish between the genuine pathogens, the microbes actually causing the infection, and those that are merely harmless colonizers, or even just contaminants from the environment or the collection process.
Right, because not everything growing there is necessarily the problem.
Precisely. This often raises an important question for microbiologists. How do you know what truly matters in that complex sample? A swab from a sore throat might contain many common bacteria, but it's crucial to identify if Streptococcus pyogenes is there, because that's the one causing strep throat. Without cultivation and differentiation, it's like looking for a needle in a haystack, blindfolded.
So once you've successfully isolated them and differentiated the culprits from the bystanders, that's when you can finally give them a name, right? It's like putting a face to the name of the microbial suspect.
That's a good way to put it. That brings us to the third purpose: identification and characterization. Obtaining sufficient growth from cultivation is absolutely essential for definitive identification and for characterizing the specific traits of those clinically relevant bacteria.
You need enough of them to actually test, I suppose.
Exactly. You simply can't perform reliable tests, biochemical assays, or even modern genomic sequencing on a single cell. You need millions, sometimes billions of them. A visible colony contains millions.
Wow.
Building on that, the fourth purpose is property demonstration. Beyond just knowing what it is, cultivation allows us to actually demonstrate specific biochemical, antigenic, and other phenotypic properties of those isolated colonies. Does it produce a particular enzyme? Does it break down specific sugars? Does it react with a particular antibody?
And these properties tell you more about how it behaves.
Yes, these aren't just academic questions. They're crucial details for understanding how that microbe causes disease and how we might combat it. It tells you about its virulence, its potential weaknesses.
Okay, this is where it gets incredibly practical and directly impacts how doctors treat patients. This seems like one of the most immediately vital applications, wouldn't you say?
Absolutely, without a doubt. The fifth purpose, antibiotic susceptibility testing, is indispensable in clinical medicine. Cultivation provides the living, growing bacteria that allow us to determine how susceptible an isolated bacterium is to various antibiotics, bacteriophages, or other antimicrobial agents.
So you test the actual bug from the patient against the drugs.
Precisely. This directly informs the most effective treatment strategies. Without cultivation and susceptibility testing, doctors would literally be guessing which antibiotic to prescribe, leading to far less effective treatments, higher patient mortality, and crucially, a significant acceleration of antibiotic resistance, which is a major global health crisis.
Right, just throwing antibiotics at things fuels resistance.
It absolutely does. So it's not just about treating this infection, but also about gathering the raw material to prevent future ones or develop better defenses for the population.
That's a perfect way to put it. And it leads us neatly to the sixth purpose.
Yeah.
Antigen or vaccine preparation. Cultivation is the fundamental groundwork for preparing antigens used in diagnostic tests. These help identify current or past infections in a patient, maybe through antibody detection. And crucially, it's how we produce the massive quantities of microbial material needed for developing vaccines to protect entire populations against diseases. Imagine trying to make a polio vaccine without being able to grow the polio virus.
Impossible, yeah.
Finally, for the very advancement of science itself, the seventh purpose is stock culture maintenance. Research labs, diagnostic labs, and even teaching institutions need to keep living collections of known bacteria. Cultivation enables the maintenance of these vital stock cultures, acting as a living library of microbes for ongoing research, quality control, testing new methods, and education.
A living library, I like that.
It's astonishing to think about how far we've come. It wasn't always as precise as it is today. Can you imagine trying to identify a single type of microbe when they're all swimming together in a murky broth? It must have been like trying to pick out a single specific grain of sand from a stirred-up beach. Utterly impossible.
Yeah, the early days were messy. Louis Pasteur, for example, a giant in the field, initially used liquid media, simple broths like urine or meat broth, basic stuff.
Urines? Really?
Well, it's sterile in the bladder and contains nutrients, but the monumental difficulty lay in isolating pure cultures from these mixed liquid environments. This, understandably, created a huge bottleneck in early bacteriology.
Because you couldn't be sure what you were studying.
Exactly. If you couldn't get a pure culture, you couldn't be certain what you were actually studying or if your results were truly attributable to one organism. It was a scientific quagmire.
Enter Robert Koch.
Precisely. And that's where Robert Koch, another titan of microbiology, stepped in. He recognized this fundamental problem and began pioneering the use of solid media. His early attempts were, let's say, inventive, given the technology of the time.
I'm sensing a butt coming.
He tried culturing bacteria on sterile, cut, boiled potatoes. While commendable for its ingenuity, it proved unsatisfactory due to incredibly inconsistent growth. Potatoes, naturally, vary wildly in their nutrient content, they spoil quickly, and they certainly don't provide all the specific nutritional requirements for every type of microbe.
Right, so a noble attempt, but probably not the most reliable foundation for a whole new science. He must have moved on from potatoes pretty quickly.
He did. Koch then experimented with solidifying liquid media using gelatin. This was a definite improvement over potato slices, offering a more standardized base. Finally, something potentially reproducible.
Okay, better. Still not perfect.
Still not perfect. It came with two significant flaws that severely limited its utility. First, many common bacteria possessed enzymes that could actually digest the gelatin, causing the solid medium to liquefy again, completely defeating the purpose of providing a stable surface for isolation.
Ah, so the plate would just turn back into soup.
Pretty much. And second, and crucially, gelatin melts at temperatures above 28 degrees C. This severely restricted its use, especially for human pathogens that thrive at body temperature, typically around 37 degrees C. So you couldn't reliably grow many of the bacteria causing human diseases. The search for a better solidifying agent was definitely still on.
This is where the story gets really good, I think. The breakthrough moment.
It really is. If we truly connect this to the broader picture of scientific progress, the real game-changing breakthrough, a moment of pure genius in hindsight, arrived in 1882. And it wasn't from Koch himself initially.
Who was it?
Fanny Iochemius Hess, the often unsung hero and wife of one of Koch's assistants, Walter Hess. She suggested using agar, something she used in her kitchen for making jellies, as a potentially superior solidifying agent.
From the kitchen to the lab. Amazing.
What's truly remarkable here is that agar, derived from seaweed, possesses properties that completely overcame gelatin's limitations. It's generally not attacked or digested by the vast majority of bacteria, ensuring the medium remains solid. That was huge.
Okay, so it stays solid. What about temperature?
And crucially, it remains solid up to a much higher temperature, around 100 degrees C, but melts near boiling and solidifies around 40-45 degrees C. This discovery was nothing short of monumental.
Why was that temperature range so important?
It meant microbiologists could now reliably grow bacteria on a stable, non-degradable solid surface at the optimal temperatures required for human pathogens, like 37 degrees C. Plus, they could melt it, sterilize it, cool it down enough to add heat-sensitive things like blood, and then pour it. It transformed bacteriology overnight.
So once they had this incredible solidifying agent, they still needed a proper, consistent container for it, right? You couldn't just pour it onto the benchtop. And that's where another familiar name in every biology classroom comes into play.
You got it. Richard Petrie, another of Koch's assistants, developed the now ubiquitous Petri dish specifically for these new solid culture media. A simple, shallow, lidded dish.
So simple yet so essential.
Precisely. These two advancements, the stable, non-degradable agar and the perfectly designed, stackable Petri dish together, enabled the truly reliable isolation of pure cultures for the very first time. It's difficult to overstate the impact of these deceptively simple yet profound innovations.
Agar and the Petri dish. The power couple of early microbiology.
Absolutely. They significantly stimulated progress across bacteriology and truly laid the essential groundwork for modern microbiology as we know it today, allowing for precise, reproducible experiments.
Okay, so now we have the solid base, thanks to agar and the Petri dish. But what actually goes into these dishes to feed the microbes? What's literally on the menu for these microscopic gourmands? And how do we ensure it meets their incredibly diverse dietary needs?
Right, the actual food. Well, the most fundamental ingredient, and one often overlooked because it seems so basic, is simply water. Surprisingly, tap water is frequently suitable for many media preparations, though purified or distilled water is often used for consistency in research.
Can't live without water, even microbes.
Nope. Then, of course, there's the star of the show, the one that genuinely revolutionized the field, agar itself.
The magic seaweed jelly.
Indeed. Agar is the primary solidifying agent. As we touched upon, it's obtained from various types of red seaweed and is primarily composed of a long-chain polysaccharide, alongside some inorganic salts, protein-like materials, and trace amounts of fatty acids.
But its real trick is the temperature thing, right?
Yes, its crucial thermal properties are its true secret weapon. It melts around 90-100 degrees C, which is hot enough to ensure thorough sterilization and allow for complete mixing of nutrients. But here's the genius part. It then solidifies at a much lower temperature, around 40-45 degrees.
And that 50-degree window is key.
It's a microbiologist's superpower. It means we can create a completely sterile base by boiling, and then without killing off delicate, crucial nutrients like blood or serum or certain vitamins that would be destroyed by high heat, we can carefully mix them in just before the agar sets. This thermal sweet spot single-handedly transformed what microbes we could successfully grow.
Clever. So you've got the water and the agar providing the structure. What about the actual food, the carbohydrates, proteins, and vitamins the microbes need to grow? Where do those come from?
That's where the growth-enriching constituents come in. These are the core nutrients that provide everything a microbe needs. Carbohydrates for energy, proteins or amino acids for building blocks and enzymes, inorganic salts like phosphates and sulfates, essential vitamins, and various growth factors.
What are common examples? Like, what do you actually put in?
Common examples include things like yeast extract and meat extract, or peptones, which are partially digested proteins. These provide a very rich general-purpose diet for a wide range of bacteria, often described as a microbial multivitamin. They cover a lot of bases.
And then for the pickier eaters, the ones that need a bit more luxury or a very specific complex blend of nutrients. You can't just put them on a basic diet, you said.
Exactly. For those truly fastidious organisms, and when we say fastidious, think of them like the gourmet eaters in the microbial world. They don't just need food, they need a very specific, rich, and often complex blend of nutrients that simpler media just can't provide. We often add blood or serum.
Actual blood.
Yep. Typically, defibrinated horse or sheep blood in concentrations of 5% to 10% or serum are used to enrich the media. These additions provide specific growth factors like hemin and NAD, enzymes, and nutrients that these demanding organisms either cannot synthesize themselves or require in specific forms. Blood agar is one of the most common media used in clinical labs.
Okay, so the media can get quite complex.
Oh, very. This raises an important question. Since microbes have such incredibly diverse needs, how do we classify and meticulously choose the right medium for a particular purpose? Laboratories don't just have one universal media. That wouldn't work.
Right, you need different tools for different jobs.
Exactly. Media are cleverly classified in several ways, each designed for a specific purpose, allowing scientists to pinpoint exactly what they're trying to grow or even trying to inhibit and why.
How are they classified then?
First, we classify them quite simply based on their physical phase. You have liquid media, often referred to as broth. These are essentially nutrients dissolved entirely in water, no agar or very little.
And how do you know if anything's growing?
Bacterial growth in broth is usually indicated by turbidity, or a visible cloudiness, as the organisms multiply and suspend throughout the liquid. Their main advantage is incredibly high sensitivity for isolating very small numbers of bacteria. A single cell can multiply throughout the entire volume, making it easier to detect.
The disadvantages too.
Definitely. It's incredibly difficult to identify mixed cultures without subculturing them onto solid media because everything's just mixed together. And it's hard to precisely quantify the amount of growth. Examples include nutrient broth, peptone water, and various enrichment media we'll talk about.
And then the opposite of that, the one that really allowed pure cultures to happen, would be solid media.
Right. Solid media typically contain about 1.5 to 2% agar, providing a firm, gel-like surface. Their immense advantages are that they allow for the growth of discrete, isolated colonies, which is absolutely critical for separating different species.
There's dots on the plate.
Exactly. They also allow for straightforward quantification. You can actually count the colonies to determine the bacterial load and identification based on colony morphology, meaning their unique size, shape, color, and texture. Classic, widely used examples include blood agar, chocolate agar, and McConkey agar.
Is there anything in between liquid and solid?
There is. Finally, there's an in-between category, semi-solid media. These have a significantly reduced agar concentration, usually only 0.20–0.4%, just enough to give it some body but still be quite soft.
What's the point of that?
Their primary purpose is quite specific, to facilitate the spread of motile bacteria within the medium, making them incredibly useful for observing bacterial motility, whether they can swim or not. Fletcher's medium for Leptospira is a good example, allowing these spiral-shaped motile bacteria to swim through the soft agar. You can see the growth spreading out from the inoculation line.
Okay, so classification by physical state. Liquid, solid, semi-solid. What's another way?
The second broad way we classify media is based on nutritional factors, reflecting the varying metabolic appetites of microbes. First, they're simple media, often called basal media.
Simple meaning?
Meaning they contain only basic nutrients and are designed for ordinary organisms, those that aren't particularly fussy about their diet and can synthesize most of what they need. They are general purpose and often serve as a foundational base for preparing more complex or enriched media. Peptone water, nutrient broth, and nutrient agar are all classic examples of simple media. They're like the plain bread and water of the microbial world.
And then I'm guessing it gets a little more complex for the more demanding microbes. You add more stuff.
Indeed. Next up are complex media. Their exact chemical composition varies slightly between batches, simply because they often use extracts from biological materials like yeast or meat or enzymatic digests of proteins, where the precise chemical breakdown isn't fully known or standardized down to the microgram.
So you don't know exactly what's in there?
Not precisely, molecule by molecule. But don't let that fool you. Most common laboratory media fall into this category because they are highly nutritious and support a wide range of microbial growth, even if the exact molecular components aren't perfectly defined. They provide a rich, ready-made buffet for a broad spectrum of microbes. Nutrient agar and blood agar are technically complex media.
This is where it really comes down to scientific precision, though. For some cutting-edge research, you need to know exactly what's in your microbial meal, right down to the last molecule.
You're spot on. That's where synthetic or chemically defined media come into play. With these, the exact chemical composition is known with absolute certainty, down to every single salt, sugar, amino acid, vitamin, and trace element. Every component is a pure chemical.
Why go to all that trouble?
This level of precision is absolutely crucial for specific research questions, such as studying a microbe's precise metabolic pathways, or determining its exact nutritional requirements, or even investigating how subtle changes in environment affect gene expression. For carbon sources, scientists might use pure glucose or glycerol. For inorganic nitrogen sources, ammonium salts or nitrates are common. Dubow's medium with tween 80 is an excellent example of a chemically defined medium, specifically formulated for the precise study of mycobacteria.
Right, for when close enough isn't good enough.
Exactly. Moving beyond basic nutritional needs, we enter the realm of special media, which are truly custom cocktails designed for very specific needs or to achieve particular, often complex, goals in microbial identification and isolation.
So these are the highly specialized designer menus for the microbial world, the stuff beyond just basic growth.
That's a great way to think about it. First among these are enriched media. These are invariably solid media. They are meticulously prepared by adding specific substances like blood, serum, or egg yolk to a basal medium like nutrient agar.
Why add those specific things?
The purpose is to support the robust growth of those fastidious bacteria we talked about, those highly demanding organisms with exacting nutritional requirements that simply won't grow or grow very poorly on simpler, less rich media. Classic examples include blood agar, which is crucial for many pathogens like streptococcus that require blood components, chocolate agar.
Wait, actual chocolate?
Ha! No, sadly not. It's actually blood agar that has been gently heated to lyse or break open the red blood cells. This releases even more growth factors like factor V, NAD, and factor X, hemin, making it suitable for extremely delicate organisms like Haemophilus influenza and Neisseria gonorrhea. The heating turns the blood brown, hence the name.
Ah, okay. Less exciting than actual chocolate.
Afraid so. Other enriched media include Loeffler's serum slope, which uses serum and is good for Corynebacterium diphtheria, and Lowenstein-Jensen LJ medium, which contains egg and is specifically used for culturing Mycobacterium tuberculosis, a very slow and demanding grower.
Okay, so enriched media are basically basal media with goodies added for picky eaters. What about enrichment media? Sounds similar.
It does sound similar, and it's often a point of confusion for students, but they're quite different. What's truly fascinating here is that enrichment media are typically liquid media designed specifically for selection, but in a subtle way. They don't just add nutrients, they contain substances that selectively stimulate the growth of certain desired bacteria while actively suppressing others in the mix.
So it gives the one you want a head start.
Precisely. They're incredibly useful for isolating desired pathogenic bacteria from clinical specimens that have a very high background of other, often harmless, microbes, like a complex stool sample. You're trying to find a needle in a microbial haystack.
Give me an example.
Okay, selenite F broth and tetrathianate broth are used to enrich for Salmonella typhi and Shigella from feces. These broths inhibit many normal gut bacteria, like E. coli, but allow the Salmonella and Shigella to multiply. You cultured the stool in the broth first, then plated out. Alkaline peptone water, APW, and Monser's terephthalate tellurite peptone water are used specifically for Vibrio cholerae, giving it a competitive advantage due to its tolerance for high pH and specific inhibitors.
Got it. So enrichment is liquid, selective growth boost. Now, this is where the media isn't just feeding them, it's actively inhibiting unwanted microbes while strongly promoting the growth of desired ones, thanks to specific chemicals added to solid media. This must make the isolation process dramatically more efficient and act like a chemical filter in the lab. These are selective media.
They are indeed powerful detective tools in the lab, usually solid media that incorporate inhibitory substances. For example, McConkey agar is widely used for Enterobacteriaceae, the gut bacteria family. It contains bile salts and crystal violet dye.
What do those do?
They are potent inhibitors of most gram-positive bacteria, thus selectively allowing gram-negative enteric bacteria like E. coli, Salmonella, Shigella to grow. It also ingeniously contains lactose and a pH indicator, neutral red.
Why the lactose indicator?
So lactose fermenters like E. coli will produce acid, lowering the pH and causing their colonies to turn distinct pink or red. Non-fermenters like Salmonella or Shigella remain pale or colorless. This immediately gives you a differential clue about their metabolic activity.
So it's selective and differential.
Clever.
Very clever. Another excellent example is mannitol salt agar, MSA. This medium contains a very high concentration of salt, about 7.5%.
That sounds harsh. It is. It inhibits almost all bacteria except for halotolerant or salt-tolerant ones, particularly Staphylococcus species. Crucially, Staphylococcus aureus, a major pathogen, is not only halotolerant but can also ferment the sugar mannitol, which is also in the agar. This fermentation produces acid, causing the pH indicator, phenol red, in the agar to turn yellow around its colonies. Other staph species that don't ferment mannitol grow, but the agar stays pink. It provides a clear differential signal for S. aureus.
So you're not just growing them, you're immediately getting a visual clue about what they are and how they behave, which speeds up identification significantly.
Absolutely. Saves a lot of time and effort. Deoxycholate citrate agar, DCA, is another selective medium, specifically designed for isolating Shigella spp and Salmonella spp from feces by using citrate and deoxycholate to inhibit coliforms and gram-positives. Thiosulfate citrate bile salt sucrose, TCBS agar, is highly selective and differential for Vibrio cholerae and other Vibrios. The high pH and bile salts inhibit most other bacteria, and V. cholerae ferments sucrose, producing large, distinctively yellow colonies.
And what about that slow grower, tuberculosis?
Right. For the notoriously slow-growing Mycobacterium tuberculosis, we use Lowenstein-Jensen LJ medium. This is enriched with egg, but it also contains malachite green dye. This dye acts as a selective agent, inhibiting many faster-growing contaminating bacteria commonly found in sputum samples, allowing the slow-growing Mycobacterium to eventually produce its characteristic rough, tough, and buff colonies.
How long does that take?
It takes patience, usually 2 to 6 weeks of incubation, a true test of a microbiologist's dedication.
2 to 6 weeks for a result. Wow. Are there others?
Oh, yes. Other highly specialized selective media include Thayer-Martin medium, which is essentially chocolate agar plus specific antibiotics, vancomycin, colistin, nystatin, sometimes trimethoprim, to inhibit normal flora and allow the isolation of delicate pathogens like Neisseria gonorrhea and Neisseria meningitis. Blood tellurite agar, also known as McLeod's or Hoyle's tellurite blood agar, is tailored for Corynebacterium diphtherio. It contains potassium tellurite, which C. diphtheria reduces to metallic tellurium, producing distinctive gray-black colonies. And crystal violet blood agar specifically uses crystal violet to inhibit Staphylococcus and select for Streptococcus pyogenes. Each of these is a finely tuned instrument for a specific microbial target, designed to bring the right microbe out of the crowd.
Okay, building on that idea of visual clues, we have indicator or differential media. You mentioned some already, like McConkey and MSA. These are fascinating because they contain a specific indicator that changes color or allows for another recognizable change when a specific bacterium grows. This helps microbiologists differentiate colonies based on their metabolic activity rather than just their physical appearance. So they're not just growing them, they're literally watching their behavior in real time via color changes.
That's a perfect description. Blood agar, which we mentioned earlier as an enriched medium, is actually also a brilliant differential medium. It distinguishes between hemolytic and non-hemolytic bacteria by the presence or absence of clear or greenish zones around the colonies.
Hemolytic meaning?
Meaning they can lyse or break down red blood cells hemolysis. Alpha hemolysis shows partial lysis with a greenish zone, like Streptococcus pneumonia. Beta hemolysis shows complete clearing, like Streptococcus pyogenes. And gamma hemolysis means no lysis at all. This is a crucial virulence factor for many pathogens and a key identification feature.
So blood agar pulls double duty, enriched and differential.
It does. Another example is Nagler's medium, which contains egg yolk. It helps identify Clostridium perfringens by indicating its lecithinase activity. This enzyme breaks down lecithin in the egg yolk and creates a distinctive opalescent sheen or precipitate around the colonies.
Very specific reactions. Okay, now, this brings us to another critical challenge in microbiology. How do you get a fragile, potentially sensitive sample from a patient, maybe in a rural clinic, to the diagnostic lab miles away without the microbes dying off, or, conversely, overgrowing and changing the original balance?
Ah, yes. Sample transport. Hugely important and often overlooked. That's where transport media come in. These are meticulously designed, usually semi-solid and non-nutritive media, intended to maintain the viability of delicate organisms in clinical specimens during transport without allowing them to multiply significantly.
Why not let them multiply? Wouldn't that make them easier to find?
Because you want the lab result to reflect the situation in the patient at the time of collection. If you let fast-growing bacteria multiply during transit, they could overwhelm slower-growing pathogens, giving you a false negative, or just change the relative proportions, giving a misleading picture. The goal is preservation, not growth. They typically contain only buffers to maintain pH and salts to prevent osmotic lysis, maybe some reducing agents for anaerobes, but deliberately lack growth factors like carbon and nitrogen sources.
So they're essentially microbial holding cells, keeping the status quo until the sample can be properly analyzed. Examples.
Stewart's transport medium is a classic, often used for preserving Neisseria gonorrhea, which is a very sensitive bacterium that dies quickly outside the body. Kerry-Blair medium is widely used for preserving enteric pathogens like Vibrio spp, Salmonella, and Shigella spp in stool samples until they reach the lab. And Pike's medium is specifically designed for transporting specimens containing Streptococcus.
Okay, that makes sense. And then we have sugar media, which are all about what the bacteria eat and how they process it, giving you vital clues about their identity based on their fermentation reactions. You mentioned McConkey already.
Exactly. These are typically simple broth or agar media containing a specific carbohydrate, like glucose, lactose, sucrose, and a pH indicator. If the bacterium ferments the sugar, it produces acid, changing the indicator color. Observing which sugars an isolate can or cannot ferment is a cornerstone of classical bacterial identification. An example is Hiss's serum peptone water medium, used for testing fermentation reactions of organisms like C. diphtheria.
And finally, for a very specific type of microbe, the oxygen haters, we have reducing media.
Yes, these are crucial because they chemically remove molecular oxygen from the environment, making them absolutely essential for the growth of obligate anaerobes bacteria that cannot survive and are often killed in the presence of oxygen. Cooked meat broth, CMB, is an excellent example, as we'll discuss more later. It contains meat particles with natural reducing agents. Another is thioglycolate broth.
Okay, quite a range of specialized menus. So you've gone through all that meticulous effort to grow your pure culture, identify it, characterize it. How do you actually keep it viable for the long term?
Yeah.
You can't just leave these living libraries out on the counter. They're like precious biological archives.
Absolutely not. Labs need to preserve these valuable cultures for future use, whether for ongoing research, quality control, vaccine development, or just having reference strains. The simplest method, suitable for short-term storage, typically for days or a few weeks, is basic refrigeration, usually around 4 degrees C. Lowering the temperature significantly slows down their metabolism, putting them in a kind of suspended animation.
But for really long-term storage, like years or even decades, you need something far more robust and stable, don't you? Something that essentially freezes time for the microbes.
You're right, refrigeration only works for so long. For truly long-term storage, labs utilize deep freezing. Pure cultures are rapidly frozen at very low temperatures, usually ranging from -50 degrees C down to -95 degrees C in specialized freezers, or even in liquid nitrogen at -196 degrees C.
You just freeze the broth?
Not usually. They are typically suspended in a cryoprotective liquid, often broth, containing glycerol or DMSO. Think of it as a special antifreeze for cells. This protects the delicate cells from damage caused by ice crystal formation during the freezing and subsequent thawing process. These meticulously frozen cultures can then be thawed and cultured years, even decades later, retaining their viability and genetic integrity. It's like putting them into a cryogenic sleep.
And there's an even more sophisticated way, right? Freeze drying.
Yes, what's truly fascinating here is perhaps the most sophisticated and longest-term preservation method available. Lyophilization, more commonly known as freeze-drying, it's the gold standard.
How does that work?
In this highly controlled process, a suspension of microbes, often with cryoprotectants, is first quickly frozen to even lower temperatures, typically ranging from -54°C to -95°C. Then, water is removed from the frozen sample by applying a high vacuum. This causes the ice to turn directly into water vapor without passing through a damaging liquid phase, a process called sublimation.
So you're basically removing all the water while it's frozen solid.
Exactly. This precisely controlled dehydration preserves organisms in a viable, dormant state for many, many years, often beyond decades, stored as a dry powder, often in sealed glass ampoules, sometimes even at room temperature. Crucially, this method almost completely avoids the formation of large destructive ice crystals that could physically damage the delicate cell structures, making it the best method for maintaining bacterial viability and genetic stability over extended periods. It's essentially creating a living fossil you can rehydrate.
That's truly incredible. The ability to preserve these living libraries, these snapshots of microbial life, is absolutely crucial for ongoing research, for the continuous development of new drugs, and for understanding how infectious disease outbreaks evolve and spread over vast stretches of time. It's like having a time capsule of microbial life that you can open and reactivate whenever you need it.
Precisely. It underpins reference culture collections worldwide.
Now that we've thoroughly explored the incredibly diverse and specialized menus we offer to microbes, let's unpack the how. The actual hands-on methods and techniques used to get them to grow on those media. We're moving from the ingredients to the art and science of the culinary preparation itself, if you will.
Okay, the practical techniques. Absolutely. First up is arguably the workhorse of virtually every microbiology lab around the globe. Streak culture, often called surface plating or just streaking.
I've seen images of those beautiful plates with the distinct separate colonies, almost like a microscopic garden. It looks quite artistic.
It can be, with practice. Streak culture is routinely employed for obtaining discrete, isolated colonies of bacteria from clinical specimens or mixed cultures, which, as we discussed, is absolutely crucial for studying bacterial properties and, most importantly, for achieving pure cultures. Without isolated colonies, it's practically impossible to know if you're working with one organism or a dozen different ones, rendering your results unreliable.
So how is it actually done? How do you get those isolated colonies?
The method involves using a sterile instrument, traditionally a platinum or nichrome wire loop, although disposable plastic loops are common now. You dip the loop into your sample, the inoculum. Then you meticulously spread that small inoculum thinly over the surface of a solid agar plate.
Just spread it all over.
No, not randomly. It's done in a specific pattern, usually dividing the plate mentally into quadrants or sections. You make close parallel streaks in the first section. Then, the key here is that the loop is flamed to sterilize it, killing any bacteria left on it, and then cooled by touching an uninoculated part of the agar briefly.
Why sterilize between sections?
To dilute the bacteria. After sterilizing and cooling, you drag the loop through the end of the first set of streaks just once or twice, pulling a much smaller number of bacteria into the second section, and then make streaks throughout that section. You repeat this process, sterilize, cool, drag from the previous section, streak for maybe 3 or 4 sections.
Ah, so each section has fewer bacteria dragged into it than the last?
Precisely. This progressive sterilization and dilution of the inoculum with each set of streaks is paramount to getting individual cells to land far enough apart on the agar surface to grow into well-separated distinct colonies on the final, most dilute streaks. After inoculation, plates are always incubated inverted, meaning lid side down.
Why inverted?
To prevent condensation from forming on the lid and dripping onto the agar surface. Those drips could cause colonies to run together and ruin the isolation.
Smart. And the outcome of all that meticulous technique, you get your beautiful, pure, and neatly separated colonies, hopefully.
Exactly. Confluent or solid growth typically occurs at the primary inoculation site, the first section, where the inoculum was most concentrated. But as you move towards the end of the streaks, where the inoculum was progressively diluted, the growth thins out, revealing those beautiful, distinct, isolated colonies that microbiologists rely on for all subsequent work. It's a foundational skill, requires practice to get right.
Next, we have lawn culture, sometimes called carpet culture. This sounds like it creates a much more widespread, uniform growth across the entire plate, not isolated colonies.
Yeah.
What's its primary purpose, given that streak culture is for isolation?
You're right. The goal is the opposite of isolation. Its purpose is precisely to provide a uniform, even layer, a lawn of bacterial growth across the entire surface of a solid medium. No gaps, just solid growth.
Why would you want that?
This method is incredibly useful for specific, high-volume applications where you need a consistent bacterial layer covering the whole plate. The most common use is antibiotic sensitivity testing, particularly the Kirby-Bauer disc diffusion method. Small paper discs impregnated with different antibiotics are placed on this uniform lawn of bacteria.
And you see how well they work.
Exactly. You incubate the plate, and if the antibiotic is effective, it will diffuse into the agar and inhibit bacterial growth, creating a zone of inhibition around the disc. The size of the clear zone tells you how susceptible that bacterium is to that specific antibiotic. It's also used for bacteriophage typing, where drops of virus suspension, bacteriophages, are placed on the lawn to see if they can infect and slice the bacteria, creating clear spots. And it's used for preparing large amounts of bacterial antigens and vaccines, where a substantial, consistent amount of bacterial growth is required from the entire plate surface.
So how do you ensure such an incredibly even layer without just getting clumps of growth like in the first part of a streak plate?
Good question. The method is designed for uniformity. The surface of a solid media plate is either briefly flooded with a standardized liquid culture or suspension, with any excess then carefully pipetted off. Or, more commonly, a sterile cotton swab is dipped into the liquid culture, excess fluid pressed out against the side of the tube, and then the swab is used to meticulously inoculate the entire plate surface evenly, going back and forth and rotating the plate to ensure complete coverage right to the edges. The plate is then simply incubated to allow the uniform lawn to develop.
Then there's stroke culture. What's its primary use, and how does it differ from a streak or a lawn? It sounds less intense.
It is less intense and usually done in a tube, not a plate. Stroke culture provides pure growth of bacteria primarily for specific diagnostic tests like slide agglutination, where a clean, concentrated sample of bacteria is needed quickly, maybe for mixing with antibody regions on a slide. You don't need isolated colonies, just readily accessible pure growth.
And how is it done?
It's performed in tubes containing nutrient agar slopes. That's agar that has solidified at an angle within the tube, providing an inclined surface, maximizing the surface area in the tube. The surface of this agar slope is lightly smeared or stroked with an inoculation lute containing the bacteria, usually in a gentle zigzag pattern from bottom to top, taking care not to break or cut into the agar surface itself. This yields a relatively thin, accessible band of pure growth on the slanted surface for rapid testing.
And stab culture. That one sounds quite literal, a direct stab into the medium. What's that for?
It is indeed literal. Stab culture is primarily used for maintaining stock cultures of certain bacteria, as inoculating deep into the agar minimizes exposure to air and reduces dehydration, helping to preserve the microbes longer, especially non-spore formers. It's also an excellent method for demonstrating the oxygen requirements of bacteria.
How does it show oxygen requirements?
Because their growth pattern along the stab line directly indicates their oxygen preference. Strict aerobes grow only at the very top surface. Strict anaerobes grow only deep down at the bottom, away from oxygen. Facultative anaerobes grow all along the stab line. Microaerophiles grow in a band just below the surface where oxygen levels are lower but not absent. Additionally, if the medium contains gelatin, it can show gelatin liquefaction. If an organism produces gelatinase enzymes that digest gelatin, the solid medium will turn liquid along the stab line.
And the technique?
The method involves taking a long, straight inoculation wire, not a loop, touching it to a colony, and carefully stabbing it deeply, maybe 2/3 of the way down, into the medium in a tube, which is often solid or semi-solid agar, solidified upright, not sloped. The wire is then carefully withdrawn along the exact same line of insertion to avoid splitting or cracking the medium, ensuring a clean inoculation track.
Okay, next up, pour plate culture. This one sounds like a much more quantitative method, perhaps for counting the actual number of bacteria rather than just isolating or testing properties.
Precisely. The primary purpose of pour plate culture is to provide a very accurate estimate of the viable bacterial count in a liquid suspension, how many living colony-forming units, CF USERs, are present per milliliter. It's a recommended method for quantitative urine cultures, for example, which is crucial for diagnosing urinary tract infections by determining the exact concentration of bacteria per milliliter of urine. A certain threshold number indicates infection.
How does pouring help count them?
The method starts with preparing appropriate serial dilutions, like 1.0, 1.10, 1.1000 of the original liquid sample, say urine or water. A measured volume, typically 1 milloa or 0.1 milloa of one or more of these dilutions, is then pipetted into an empty, sterile Petri dish. Then you add molten solid agar medium that has been carefully cooled to around 45 degrees C.
Cooled agar.
Yes, it has to be warm enough to still be liquid for pouring, but cool enough not to kill the heat-sensitive bacteria when they are mixed in. You swirl the dish gently to mix the bacteria evenly throughout the molten agar. This mixture is then allowed to solidify.
So the bacteria are trapped inside the agar.
Exactly. What's unique and advantageous about this method is that colonies will grow both within the agar itself, as small, often lens-shaped colonies, and on the surface. You count all the colonies on a plate from a dilution that gives you a countable number, usually 3,300 colonies, and then multiply by the dilution factor to get the original concentration. It's considered more accurate for counting than just spreading on the surface sometimes.
And what about shake culture? Does that involve, well, shaking the culture vigorously?
Not continuous shaking during incubation, no, but it does involve mixing the inoculum thoroughly at the start. Its purpose is primarily visual to observe the growth patterns of organisms relative to oxygen concentration within the medium, similar to a stab culture, but perhaps clearer in a liquid medium before it solidifies.
How's it done then?
It typically involves taking a tube of molten agar, inoculating it with a small amount from a liquid culture, mixing the contents thoroughly by gently rolling or swirling to distribute the bacteria evenly, and then allowing the agar to solidify rapidly, usually in an ice bath while upright. As the agar solidifies, it traps a natural oxygen gradient, with higher oxygen at the top surface exposed to air and progressively less oxygen deeper in the tube. Organisms will then grow in specific zones depending on their oxygen needs after incubation. Strict aerobes at the top, strict anaerobes at the bottom, facultators throughout, microaerophiles in a band just below the surface.
So another way to check oxygen requirements. Finally, we have liquid culture. We talked about broths earlier as a type of media, so this is about the method of inoculating them and their specific uses.
Yes, exactly. Using liquid media, or broth cultures, liquid culture is invaluable, especially when the concentration of bacteria you're looking for is expected to be very small, such as in blood cultures. Even a few bacteria in the bloodstream can indicate sepsis, a serious life-threatening condition, and broth provides the best chance of detecting them as they can multiply throughout the volume.
Any other key uses?
It's also used for sterility tests, for instance, testing pharmaceutical products or medical devices to ensure they are entirely free of any microbial contamination before release. Furthermore, it's often preferable for inocula that might contain residual antibiotics or inhibitory substances, perhaps from a patient already on treatment. These compounds are quickly diluted and potentially inactivated in the larger fluid volume of the broth, allowing even sensitive organisms that might be inhibited on solid media to grow.
And how do you inoculate a broth?
It's usually simpler than plating. Liquid media in tubes, flasks, or bottles are inoculated by simply touching the surface or dipping the loop into the broth with a charged loop, a loop that has picked up bacteria from a colony or sample, or by adding the inoculum precisely with sterile pipettes or syringes, for example, when adding blood to blood culture bottles. A key point to remember, though, is that while liquid cultures are excellent for initial growth and detection of low numbers, subculture onto solid media is almost always required for final identification and characterization, as it's very difficult to distinguish mixed cultures or assess colony morphology in a uniformly cloudy broth.
Right, you still need those isolated colonies eventually.
Usually, yes. Broth is often the first step in detection or enrichment. So we've talked about all these wonderful techniques in media for growing bacteria that like, or at least tolerate, oxygen. But what about those microbes that can't stand oxygen? This raises a profound question. How do you reliably grow organisms that find our atmospheric oxygen, about 21% of the air we breathe, not just unhelpful, but genuinely toxic? It's a significant engineering and biological challenge.
Right. You can't just leave them out on the bench. They die almost instantly. You need a completely sealed, oxygen-free environment. It's like trying to cultivate a deep-sea fish in the desert.
Exactly. Anaerobic cultures typically require an environment completely free of molecular oxygen, or at least reduced to very low levels. Many clinically significant anaerobes also prefer an atmosphere enriched with carbon dioxide, maybe 5–10%. They are commonly incubated at 35–37 degrees C or 95–98.6 degrees Airfo for at least 48 hours and often much longer, sometimes a week or more, as many clinically significant anaerobes grow very slowly compared to facultative bacteria.
Why is oxygen so toxic to them?
It comes down to biochemistry. Normal aerobic respiration produces some toxic byproducts, highly reactive oxygen species like superoxide radicals, O2, and hydrogen peroxide, H2O2. Aerobes and facultative anaerobes have enzymes like superoxide dismutase, SOD, and catalase or peroxidase to break these down safely. Many obligate anaerobes lack one or both of these protective enzymes. So, when exposed to oxygen, these toxic molecules build up inside their cells and damage vital components like DNA, proteins, and lipids. Oxygen is literally poisonous to them.
Okay, so how do you create that oxygen-free bubble for them in the lab?
There are several methods, ranging from traditional to more modern. One widely used and historically dependable method is the Macintosh-filled anaerobic jar. This is essentially a robust, sealed glass or metal jar with an airtight lid that clamps down securely.
How does it get the oxygen out?
Inside the lid, it contains a catalyst, typically palladium-coated alumina pellets, usually held in a wire mesh sachet. The mechanism is quite clever. After placing your inoculated plates or tubes inside the jar, you seal the lid. Then, air is first evacuated from the jar using a vacuum pump connected to an inlet valve. After evacuation, hydrogen gas, or a mixture of hydrogen and CO2, is introduced through another valve. The palladium catalyst then facilitates a chemical reaction at room temperature, combining this introduced hydrogen with any residual oxygen remaining in the jar, forming water, 2H plus O2HO.
So it chemically scrubs the oxygen out using hydrogen.
Exactly. This reaction effectively scavenges all the oxygen, thereby achieving complete anaerobiosis. You often use an indicator strip inside the jar, usually methylene blue, which is blue when oxygen is present but turns colorless when anaerobic conditions are achieved, confirming the system worked. You can also often see condensation on the inside of the jar as water forms.
That's a very elegant chemical solution for creating the right environment. Sounds a bit cumbersome with vacuum pumps and gas tanks, though. Is there an easier way?
There is. For more routine use and convenience, there's the gas pack system, or similar commercial systems. It's like a disposable, self-contained anaerobic system for use in a sealed jar, often made of clear polycarbonate plastic.
How does that work without pumps and tanks?
It uses a disposable paper sachet or envelope containing chemicals. When you add a specific amount of water to activate it, or sometimes it activates just on exposure to air, the chemicals react to produce hydrogen and carbon dioxide gases. Another component in the sachet, often ascorbic acid or activated carbon, simultaneously absorbs the oxygen present within the sealed jar. You simply place the activated sachet in the jar along with your inoculated plates in an anaerobic indicator strip, seal the lid tightly, and incubate. It does the work of creating that oxygen-free, usually CO2-enriched environment chemically. It's much simpler and generally safer than handling pressurized gas tanks.
Very convenient. Now, earlier you mentioned a special broth. Here's where it gets particularly interesting with a specialized liquid medium. One's been around for a long time. Cooked meat broth, CMB, sometimes called Robertson's cooked meat, RCM medium. You said it was important for anaerobes.
It truly holds a special, almost revered place in anaerobic bacteriology because of its unique properties and long history of successful use. It's a complex medium containing nutrient broth and particles of minced meat, usually heart muscle.
Why cooked meat?
The key lies in the meat particles themselves. They contain natural reducing substances like glutathione from the muscle tissue and unsaturated fatty acids. These act as powerful chemical reducing agents, actively lowering the redox potential, essentially the oxygen tension in the medium. They chemically react with and absorb any dissolved oxygen, creating anaerobic conditions deep within the broth, especially down amongst the meat particles.
So the meat itself makes the broth anaerobic?
Largely, yes. This allows for the cultivation and robust growth of even the most strict anaerobes, those organisms that are most sensitive to any trace of oxygen. It's great for recovering small numbers of anaerobes. Furthermore, it offers a fascinating visual clue about their metabolic activities. Saccharolytic anaerobes, those that primarily ferment sugars, will typically turn the meat pink as they produce acidic byproducts. In contrast, proteolytic anaerobes, which digest proteins, will break down the meat proteins, causing the meat to turn black and sometimes disintegrate, often accompanied by gas production and a putrid odor. It gives you an early clue about the type of anaerobe growing. It's also an excellent medium for the preservation of stock cultures of anaerobes due to its long-lasting reducing properties.
So a multi-purpose broth that grows them, gives you a diagnostic hint, and preserves them.
Oh.
What about other liquid options for oxygen-sensitive microbes?
Another very important one is thioglycolate broth, often fluid thioglycolate medium, FTM. It's widely used, especially for sterility testing and cultivating a broad range of bacteria, including anaerobes. It contains sodium thioglycolate and sometimes L-cysteine.
And those are reducing agents?
Yes, they are strong chemical reducing agents. They actively react with and remove molecular oxygen dissolved in the medium. Usually a small amount of agar, around 0.075%, is also added to increase viscosity and prevent the rapid diffusion of oxygen back into the deeper layers from the surface. Often, a redox indicator like resarin is included, which is pink when oxygen is present, usually in a shallow layer at the very top, and colorless under anaerobic conditions below. This creates a clear oxygen gradient within the test tube.
So you can see where the oxygen is.
Right. Anaerobes, as you'd expect, will then grow primarily down in the colorless zone at the bottom of the tube where oxygen levels are lowest. Aerobes will be restricted to the pink layer at the top. Facultative anaerobes will grow throughout the tube, both pink and colorless zones. It's a quick visual test for oxygen requirements, as well as a good growth medium.
Okay, those are jars and broths. What about bigger scale work, or when you need to handle the cultures without exposing them?
For that, what's truly fascinating for modern dedicated anaerobic labs are the more advanced, commercially produced systems known as anaerobic cabinets or anaerobic workstations or glove boxes. These are self-contained, completely enclosed units, usually made of clear plastic or metal.
Like a sealed aquarium you work in.
Kind of, yeah. They provide a continuously maintained, strictly oxygen-free atmosphere inside, usually a mixture of nitrogen, hydrogen, and carbon dioxide, meticulously controlled by gas regulators and monitors. Oxygen is scrubbed out using catalysts, similar to the jars but on a continuous basis. The major advantage of these cabinets is that all processing steps, including opening plates for examination, performing inoculations, making smears, and subculturing, can be performed inside the cabinet. You work through built-in sealed gloves, like an incubator combined with a safety cabinet.
So the cultures never see room air.
Never. This greatly improves the recovery rate and viability of even the most fastidious and oxygen-sensitive anaerobes, like many found in the gut or in deep tissue infections, reducing the chances of losing your precious culture due to brief, accidental oxygen exposure during handling. It's the gold standard for serious anaerobic work.
So you literally stick your hands into those built-in gloves and work entirely within the sealed, oxygen-free environment. That seems like the ultimate level of control, but maybe expensive and complex.
Precisely. They are expensive and require maintenance, so they're usually found in larger clinical labs or research settings doing a lot of anaerobic bacteriology. For a simpler, often disposable option for individual plates or small batches, there are anaerobic bags or pouches.
Bags. Like Ziploc bags.
Sort of, but specifically designed for this purpose. These are commercial, single-use, often transparent plastic bags or pouches. You place your inoculated plates inside, add an oxygen removal system, often a small activated sachet similar to those used in gas pack jars that generates H2 and CO2 and absorbs O2, and then you heat seal the bag closed.
So a mini anaerobic jar, essentially.
Pretty much. You then incubate the sealed bag. A convenient feature is that plates can often be examined through the transparent bag without opening it and breaking the anaerobic conditions, again preventing any oxygen exposure until you're ready for final processing. They're very practical for smaller labs or for transporting potentially anaerobic cultures.
Okay, a whole suite of tools just to avoid oxygen. Fascinating. So let's switch gears slightly. You've got your initial sample, perhaps it's a clinical specimen or maybe an environmental sample like soil or water. It's almost certainly a mixture teeming with different kinds of microbes. How do you perform that ultimate microbial separation act to get just one single type of pure culture for study? This is the art, and indeed the science, of achieving true purity in a culture, which we said was vital.
Indeed. The fundamental goal here is to obtain a pure culture, a population of microorganisms derived, theoretically, from a single parent cell. This ensures that any tests you do are characterizing just that one organism and not a confusing mixture. As we discussed earlier when talking about culture methods, the most routine, widespread, and fundamentally important method for this is surface plating, specifically the streak culture technique we detailed.
Because it physically spreads them out.
Exactly. It physically separates individual cells on the agar surface by progressive dilution across the plate. Each single cell, if it lands far enough apart from others and the conditions are right, will then grow and multiply over many generations into a visible, distinct, isolated colony, which is essentially a clone of the original parent cell. This makes it incredibly easy for a microbiologist to aseptically pick a single, well-isolated colony with a sterile loop or needle for further purification by restreaking, and study, ensuring you're working with only one species and not a contaminating mixture. It's like magic, but it's just meticulous dilution and careful technique.
The cornerstone method. But are there other tricks, especially if the one you want is rare in the sample?
Oh, definitely. Streaking alone might not be enough if your target is heavily outnumbered. If we connect this to the broader picture of how labs actually work, this is where the clever media designs we discussed earlier truly shine. The use of selective enrichment or indicator media is absolutely indispensable for isolating specific microbes from complex mixtures.
Right, using the media to favor your target or suppress others.
Precisely. These specialized media, as we covered, contain ingredients that either inhibit the growth of unwanted organism, selective media, or selectively stimulate the multiplication of specific species you're interested in, enrichment media, or help you visually distinguish your target colonies, differential media. Using these makes it vastly easier to isolate the desired one from a complex mixture. For instance, if you suspect salmonella in a stool sample, which is teeming with hundreds of other bacterial species, using a selective medium like McConkey or DCA or an enrichment broth like selenite F first will inhibit most of the normal gut flora and allow the salmonella to grow predominantly, or at least be more easily spotted. This dramatically simplifies the isolation process compared to trying to pick it out from a general culture plate where it might be a tiny minority, easily missed. It's a huge time saver and accuracy booster.
So using the right food is a major strategy. What about treating the sample before you even put it on the plate? So sometimes you need to pre-process the original sample itself to give your target organism a head start, almost like a pre-screening, to eliminate the noise. This is selective treatment of the specimen.
Yes, exactly. Sometimes the best approach is to modify the sample before inoculation. For example, heating a sample can be a surprisingly effective selective treatment, particularly if you're looking for spore-forming bacteria.
How does heating help?
Applying controlled heat, typically at 65-80 degrees C for a short period, like 10-30 minutes, can selectively eliminate most vegetative bacteria, which are generally heat-sensitive and will be killed. However, bacterial spores, like those produced by Clostridium or Bacillus species, are highly resistant to heat. They will survive this treatment. Afterwards, when you culture the heat-treated sample, only the spore formers will grow. This is particularly useful for environmental samples, such as soil, where you are specifically looking for these hardy, spore-forming organisms amidst a vast population of vegetative cells.
Clever. What about using chemicals on the sample first? Like when you have too much noise from harmless bacteria that are just overwhelming your target.
That's another important strategy. Pre-treatment with bactericidal substances. This involves using specific chemicals, often strong alkali, like sodium hydroxide or acid, to which most harmless commensal bacteria, the normal flora found on skin or in bodily secretions, are highly susceptible and will be killed relatively quickly. But crucially, the desired pathogen might be more resistant due to its cell wall structure or other properties.
And the classic example is?
The classic and extremely important example is the pre-treatment or decontamination of sputum samples for the isolation of Mycobacterium tuberculosis, TB. Sputum, coughed up from the lungs, is inherently full of normal bacteria from the mouth and throat, which grow much faster than the TB bacillus. Treating the sputum sample with a decontaminating agent, like N-acetyl-L-cysteine and sodium hydroxide, NaLC-NaOH method, effectively liquefies the thick mucus and kills these rapidly growing contaminants. The Mycobacterium, which has a unique, waxy, lipid-rich cell wall, is relatively resistant to this harsh treatment. After neutralization, the treated sample is concentrated by centrifugation and then inoculated onto specialized media like LJ medium. This allows the slow-growing TB bacilli to be isolated without being completely overgrown by the normal flora. It's essential for TB diagnosis.
So you selectively kill the competition before plating. Makes sense. What about using the environment itself?
Right. Beyond the media itself, manipulating the entire incubation environment can also act as an incredibly powerful selective tool. This is the use of selective growth conditions.
So specific temperatures, or controlling the air composition, for example, like we discussed for anaerobes.
Exactly. Temperature is a key factor. Incubating cultures at specific temperatures can selectively favor certain bacteria while inhibiting others. While most medically important bacteria grow well at mesophilic temperatures, around 37 degrees C, mimicking human body temperature, some pathogens might prefer higher thermophilic or lower psychrophilic temperatures. For instance, Campylobacter jejuni, a common cause of gastroenteritis, grows optimally at 42 degrees C. By incubating stool culture plates intended for Campylobacter isolation at this elevated temperature, you selectively inhibit the growth of many other enteric bacteria that prefer 37 degrees C, making it easier to isolate the Campylobacter.
And atmosphere.
Atmosphere is another critical condition. As we just discussed in detail, cultivating under strictly aerobic oxygen-rich or anaerobic oxygen-free conditions, or even microaerobic low oxygen increased CO2 conditions, can effectively separate organisms based on their oxygen requirements. If you incubate a mixed sample known to contain both aerobes and anaerobes in an anaerobic jar, you will only get the anaerobes to grow, effectively isolating them from any obligate aerobes present in the original mixture. Similarly, using a microaerobic atmosphere is crucial for isolating organisms like Campylobacter or Helicobacter pylori.
Okay, one more really neat trick. Here's a really clever technique that takes advantage of how some bacteria moves. Separation of motile from non-motile bacteria. You mentioned motility testing earlier. This sounds like using that property for isolation. It's like a microscopic obstacle course or race.
It's a brilliant example of physical separation based on biological function. Using a specialized device like a Krabbe's tube can achieve this. A Krabbe's tube is essentially a test tube containing semi-solid agar with a smaller bottomless glass tube placed inside it, open at both ends, sticking up slightly above the agar surface in the main tube.
Okay, a tube within a tube in soft agar. How does it work?
You inoculate the mixed culture into the agar inside the inner smaller tube. Now, only motile bacteria, those that can actively swim using flagella, have the ability to traverse or swim through the semi-solid agar, exit the inner tube, swim across the surface or through the agar in the outer tube, and eventually appear as growth on the agar surface of the outer tube, away from the initial inoculation site.
And the non-motile ones?
The non-motile bacteria remain confined to the area where they were inoculated, inside that inner tube, because they can't swim through the agar matrix. So, by taking a sample from the growth that appears in the outer tube, you obtain a pure culture consisting only of the motile organisms from the original mixture. It's a fantastic trick when motility is a key distinguishing feature and you want to separate movers from non-movers.
That is really clever. Using their own movement against them, in a way.
We're using it for them, depending on your perspective. It's elegant microbiology.
Wow. What an absolutely incredible deep dive into the invisible world of microbial cultivation. Seriously, from the history-making discovery of agar and the humble petri dish, which truly unlocked modern microbiology as we know it.
Absolutely fundamental discoveries.
To the incredibly specific menus or media we design, and the ingenious methods like streaking, selective treatment, and even using motility for growing, isolating, and even preserving these tiny organisms. We've seen just how much meticulous science and, frankly, creativity goes into understanding life at the microscopic level. It's truly a foundational science that underpins so much.
It really is. You now have, I hope, a pretty comprehensive understanding of how scientists manage to isolate and study single species from what often starts as a complex soup of microorganisms. It's not magic, it's method. This fundamental laboratory process underpins so much of modern medicine, from accurately diagnosing infections,
which lets doctors choose the right treatment,
to meticulously testing antibiotic effectiveness to combat resistance, to ensuring our food is safe to eat by detecting pathogens, and even developing the vital vaccines that protect us. It's a testament to human ingenuity applied to the smallest yet arguably most powerful forms of life on our planet. Without cultivation, the vast majority of our knowledge about bacteria and many other microbes would simply not exist, would still be largely in the dark.
So thinking bigger picture now, what does this all mean for us, beyond the lab bench and the clinic? Consider how these seemingly simple techniques, creating the right food and environment, separating the unseen, are not just about curing diseases here on Earth, vital as that is.
Where are you going with this?
Well, they are the very foundation for understanding microbial life in the most extreme environments imaginable, aren't they? From the crushing pressures and boiling temperatures of deep-sea hydrothermal vents to the frozen desolation of Arctic ice or hypersaline lakes. Cultivating microbes from these places pushes the boundaries.
True. Finding life in those extremes relies on adapting these cultivation techniques.
And this knowledge, in turn, directly informs our ambitious search for extraterrestrial life on other planets, like Mars or the moons of Jupiter and Saturn. It helps us understand the absolute limits and the diverse conditions under which life, as we know it, or perhaps even different from us, can persist and potentially thrive. What kind of media would an alien microbe need, right?
A fascinating question. Astrobiology depends heavily on these principles.
And perhaps, as we continue to refine these methods, especially finding ways to grow the unculturable majority, the vast percentage of microbes we see under the microscope but currently cannot grow in the lab,
which is a huge challenge.
They could unlock entirely new biotechnological possibilities. discovering novel enzymes for industry, finding microbes that can break down plastics or produce new biofuels, or even discovering entirely new classes of antibiotics from the vast, untapped microbial world that we haven't been able to culture yet. What more incredible secrets could these invisible worlds reveal if we just keep refining our methods of listening to them, truly understanding their needs, and finally coaxing them to grow for us?
The potential is immense. There's still so much we don't know because we can't yet grow it.
Thank you for diving deep with us today. We really hope you've enjoyed this journey into the fascinating, intricate, and often surprising world of microbial cultivation.
It was a pleasure. It's a world that impacts us all every single day.